How do we isolate rare immune cells? Imagine trying to find a needle in a haystack. That's what isolating rare immune cells can feel like. These cells are tiny in number but big in importance for understanding immunity. To do this, scientists use special techniques that pick out these cells from a large mixture of other cells. The key is to identify what makes these rare cells unique, like specific surface markers or physical traits. Once identified, they can be separated while keeping them alive and functional for study. One common method is called immunomagnetic separation. Scientists attach tiny magnetic beads coated with antibodies that recognize specific surface markers on the target cells. When a magnetic field is applied, the labeled cells are pulled out of the mixture. This technique is quick, gentle, and works well for large numbers of cells. It's often used to isolate T cells, B cells, dendritic cells, and monocytes. Automation makes it even faster and more consistent. Another powerful method is fluorescence-activated cell sorting, or FACS. Here, cells are tagged with fluorescent antibodies that stick to certain markers. The cells then pass through a machine called a flow cytometer. The machine detects the fluorescence and sorts the cells individually based on their marker patterns. FACS is very precise and can separate cells based on multiple markers at once. It's especially useful for very rare cells that need high purity. A newer technique is magnetic ratcheting technology. It uses tiny magnetic pillars to separate cells labeled with magnetic beads. This method can isolate rare cells even from very small samples, and it keeps the cells healthy for further testing. It's automated and efficient, making it a good choice for delicate or low-frequency cells. Buoyancy-activated cell sorting is another option. It uses microbubbles that attach to unwanted cells. These cells float away under gravity, leaving the rare immune cells behind. This method is gentle and doesn't need magnets or complex equipment, which helps preserve cell health. For tissue samples, laser capture microdissection is used. It employs a laser to cut out specific cells based on their location and markers. This is useful for studying cells in fixed or archived tissue samples, but yields fewer cells. Lastly, when dealing with very few or fragile cells, scientists can use single-cell isolation techniques. These include manual micromanipulation or microfluidic devices that pick out individual cells for detailed analysis like single-cell RNA sequencing. Choosing the right method depends on the sample type, how rare the cells are, and what kind of analysis is needed. These techniques help researchers identify and study immune cells that are hard to find but play big roles in health and disease. By targeting unique features of these cells, scientists can unlock secrets about how our immune system fights infections, responds to vaccines, or contributes to autoimmune conditions.